In the vast landscape of contemporary scientific research, few mysteries are as fascinating as the one hidden within our own body, in the microscopic depths of our DNA. The molecule that contains the instructions for our existence, the code that defines what we are, still hides secrets that modern science struggles to fully decipher. When in 2003 the Human Genome Project announced the completion of mapping our genetic heritage, many believed we would finally fully understand human nature. Yet, more than 20 years later, much of our DNA remains shrouded in mystery. The human genome consists of approximately 3 billion base pairs, but surprisingly only a minimal part, about 1.52%, directly codes for the proteins that make up our body. The remaining 98% was long labeled as junk DNA, a term that reflected the initial belief that these vast genetic regions were functionless, mere residues of evolution. Today we know that this definition was premature and probably incorrect. Non-coding DNA is anything but useless, but its functions remain largely enigmatic. In this documentary, we will explore the frontiers of knowledge about non-coding DNA, investigating both the most accredited scientific explanations and alternative theories that push beyond the boundaries of conventional science. We will delve into the debate on panspermia, the hypothesis suggesting that the seeds of life may come from deep space, and critically analyze theories that hypothesize ancient genetic engineering interventions by extraterrestrial civilizations. Our journey will cross the boundary between science and speculation, between what we know and what we can only imagine. Because ultimately, the question about our origins is perhaps the oldest and deepest that humanity has ever posed. Where do we come from? And what if the answer were written in our DNA, in that 98% that we still don't fully understand? The human genome can be compared to a vast ocean, of which we have thoroughly explored only the coasts, while the abyssal depths remain largely unknown. When the Human Genome Project was completed in 2003, after 13 years of work and an investment of about $3 billion, scientists were surprised to discover that the number of protein-coding genes was much lower than expected, about 20,000 to 25,000, a number not much higher than that of much simpler organisms, like the nematode worm C. elegans. This discovery immediately raised a fundamental question. If human complexity doesn't derive from a greater number of genes, where does it come from? The answer seems to reside, at least in part, in that 98% of DNA that doesn't directly code for proteins. As the Telethon Foundation explains, in reality, much of this DNA is transcribed into particular RNA molecules that don't code for proteins, which is why they're called non-coding, but that perform important regulatory functions. Non-coding DNA includes several categories of sequences, each with different characteristics and possible functions. Introns, for example, are portions of genes that are transcribed but then removed during the maturation of messenger RNA. They represent about 26% of the entire human genome, and contrary to what was initially thought, their removal can severely compromise the functionality of the gene, suggesting an important role in the gene expression process. Another significant component of non-coding DNA consists of repetitive elements, DNA sequences that repeat numerous times in the genome. Among these, retrotransposons of viral origin, HER-V, human endogenous retrovirus, constitute about 8% of our genome, but some researchers hypothesize that this percentage could reach up to 25%. These elements, which derive from ancient viruses that integrated their genetic material into the genome of our ancestors, have played an important role in the evolution of mammals. Some of them, for example, have been domesticated to perform essential functions, such as the formation of the placenta. Intergenic regions, which separate one gene from another, often contain regulatory sequences that control the expression of nearby genes. These sequences, called enhancers, silencers, or promoters, determine when, where, and to what extent a gene is expressed. Their complexity and variety contribute to the sophisticated regulation of gene expression that characterizes complex organisms like humans. In recent years, research has revealed the existence of a vast, submerged world of non-coding RNAs, molecules transcribed from DNA, but not translated into proteins. These include microRNAs, long non-coding RNAs, and many other types of molecules that play crucial roles in regulating gene expression, chromatin structure, and numerous other cellular processes. The discovery of these molecules has revolutionized our understanding of the genome, suggesting that non-coding DNA is anything but junk, but rather a sophisticated regulatory system. As highlighted in the Ultima Voce article, recent research conducted by Lund University and published in Cell Stem Cell suggests that the crucial differences that distinguish us from our closest relatives, chimpanzees, might reside precisely in non-coding DNA. Professor Johan Jakobsen, an expert in neuroscience, used stem cells to study the development of human and chimpanzee brains, discovering that the most significant differences are not found in coding genes, but in that 98% of DNA that for decades was considered marginal. This discovery represents a paradigm shift in genetic research. If we want to understand what makes us human, we must look beyond the 2% of coding DNA and explore the vast dark regions of our genome. As Jacobson himself states, unfortunately, or perhaps fortunately, since a physicist once said, "I am happy when we discover that we don't understand the universe. As long as there are things to understand, I have a job." In all probability, the difference doesn't reside in the 2% of coding DNA, but is hidden in the remaining 98%. Contrary to what the provocative title of this documentary suggests, there is no scientific consensus on an incomprehensible 2% of the human genome. Rather, it is the remaining 98% non-coding DNA that continues to challenge our complete understanding. However, within this vast genetic territory, there are indeed particularly enigmatic regions that resist attempts at interpretation and classification. One of the most intriguing aspects of non-coding DNA is its evolutionary conservation. Some non-coding sequences show a surprisingly high degree of conservation among different species, sometimes even among phylogenetically very distant organisms. This suggests that such sequences perform important biological functions. Otherwise, they would have been eliminated or modified during evolution. As reported on the page about non-coding DNA, some non-coding sequences show a very high conservation among numerous species, some phylogenetically very distant, a phenomenon difficult to explain if these regions were truly functionless. Another enigma is represented by the so-called C-value paradox, or C-value enigma, namely the lack of correlation between an organism's complexity and the size of its genome. The amoeba dubia, a unicellular organism, possesses a genome more than 200 times larger than the human one, while the pufferfish, Fugu rubrips, has a genome 10 times smaller than ours, despite having a comparable number of genes. The main difference seems to reside precisely in the amount of non-coding DNA, raising questions about its role in evolution and the complexity of organisms. Ultra-conserved regions of the genome represent another fascinating mystery. These are sequences of non-coding DNA that have remained practically identical for hundreds of millions of years of evolution, showing conservation superior even to that of many essential genes. Their function remains largely unknown, but their extraordinary conservation suggests a fundamental role that science has not yet fully understood. Transposable elements, or jumping genes, constitute about 45% of the human genome. These mobile genetic elements can move from one position to another in the genome, potentially altering gene expression and contributing to genetic diversity. Some of them derive from ancient viruses that integrated their genetic material into the genome of our ancestors. Their abundance and diversity raise questions about their role in human evolution and the possible external origin of part of our genetic heritage. A particularly interesting experiment, cited on the page, attempted to evaluate the importance of non-coding DNA by removing about 1% of these sequences from the genome of some mice. Surprisingly, the animals showed no evident phenotypic alterations. This result can be interpreted in two ways. Either non-coding DNA actually has no function, or the methods used were not sufficiently sensitive to detect the changes induced by its removal. The question remains open and subject to debate in the scientific community. Recent research is beginning to unveil some of the hidden functions of non-coding DNA. For example, it has been discovered that many of these regions are transcribed into non-coding RNAs, which play crucial roles in regulating gene expression, chromatin structure, and numerous other cellular processes. However, for many non-coding sequences, the function remains unknown or only partially understood. As highlighted in the Science Future article, within the human genome, there is a region that provides no instructions for producing proteins, i.e., does not code for proteins. These regions, once considered useless, are revealing unexpected potential in biomedical research, including the understanding of complex diseases like cancer. Mutations in non-coding regions can indeed alter the expression of crucial genes, contributing to the development of pathologies. The enigma of incomprehensible DNA represents one of the most fascinating frontiers of contemporary genetic research. Like a vast unexplored territory, these regions of our genome might hide fundamental secrets about our biology, evolution, and perhaps our origins. Science proceeds gradually in their decipherment, but the path is still long and full of surprises. Once all our attempts to obtain living matter from inanimate matter prove vain, it seems to me fully correct scientific procedure to ask whether life has in reality ever had an origin, whether it is not as old as matter itself, and whether spores could not have been transported from one planet to another and have taken root where they found fertile ground. With these words, the German physicist Hermann von Helmholtz expressed already in the 19th century the essence of what would become known as the theory of panspermia. Panspermia, from the ancient Greek term pansperm, meaning all seeds or common seed, is a hypothesis suggesting that the seeds of life are distributed throughout the universe and can travel through space, transported by meteorites, asteroids, comets, or stellar dust. According to this theory, life on Earth might not have been born here, but arrived from deep space, carried by these cosmic messengers. However, the theory of panspermia also presents important limitations and criticisms. The main objection is that it doesn't really solve the problem of the origin of life, but simply shifts it to another celestial body. As highlighted on the page, critics point out that the hypothesis doesn't solve the problem of how life originated, but simply shifts it to another celestial body. Moreover, despite the demonstrated resistance of some microorganisms to space conditions, interstellar travel would present enormous challenges for any form of life, including extremely long transit times and exposure to potentially lethal cosmic radiation. Interstellar panspermia, in particular, remains highly speculative. The scientific community generally considers panspermia a plausible hypothesis regarding the transfer of organic molecules or possibly microorganisms within the solar system, local panspermia, but remains more skeptical about interstellar or intergalactic panspermia. Regardless of its validity, the theory of panspermia raises fascinating questions about the distribution of life in the universe and the possibility that Earth is not an isolated biological island but part of a cosmic network of life. As Sapere Ambiente writes, what if life on Earth were alien? This question, however provocative, invites us to consider the possibility that our origins are more cosmic than we have imagined so far. Among the alternative theories on the origin of parts of our DNA, one of the most fascinating and controversial is that which hypothesizes ancient genetic engineering interventions by advanced extraterrestrial civilizations. This theory, which lies at the boundary between scientific speculation and science fiction narrative, is closely linked to the so-called ancient astronauts theory, which suggests visits and interventions by extraterrestrial beings in human prehistory. The main promoter of this view was the Swiss writer Erich von Däniken, author of numerous books on the subject, including the famous Chariots of the Gods, published in 1968. Von D. Nyken interpreted various archaeological artifacts and ancient texts as evidence of the presence of extraterrestrial visitors on Earth in remote times. According to this theory, these advanced beings would have not only influenced the cultural and technological development of primitive humanity, but might also have genetically manipulated our ancestors. Another influential author in this field was Zecharia Sitchin, who in his books proposed the existence of an alien race called Anunnaki, originating from a hypothetical planet called Nibiru. According to Sitchin, the Anunnaki would have created human beings through genetic engineering, combining their DNA with that of primitive hominids to obtain a race of slaves to employ in mining. Supporters of these theories often point to non-coding DNA as a possible signature of these ancient genetic interventions. The idea is that these vast regions of our genome, whose function remained mysterious for a long time, might contain codes or instructions intentionally inserted by advanced extraterrestrial civilizations. Some theorists speculate that so-called junk DNA could be a sort of dormant program, destined to activate under certain conditions or at specific stages of human evolution. These theories also feed on some peculiarities of human evolution, such as the rapid development of the brain and cognitive abilities that has characterized our species. According to supporters of alien intervention, this evolutionary leap would be too rapid and significant to be explained through conventional mechanisms of natural evolution. However, as highlighted in the CICAP article, Ancient Astronauts, Fant Archaeology and the Importance of Context, these theories present significant methodological problems. First, they tend to decontextualize archaeological artifacts, interpreting them without considering the cultural, historical, and artistic context in which they were produced. For example, statuettes like the Japanese dog, or the so-called astronaut of Palenque, are interpreted as representations of alien visitors in spacesuits, ignoring more plausible interpretations based on knowledge of the cultures that produced them. Second, supporters of these theories tend to select only the artifacts that seem to support their view, ignoring the vast majority that do not. As the CICAP article explains, Fant archaeologists typically show only a single specimen of the mysterious artifact, which seems to come out of nowhere and can indeed appear difficult to explain, taken in isolation. They do not cite the many other specimens that would help understand how that object developed until it assumed that particular appearance. Another methodological problem is the use of the argument from ignorance. Gaps in scientific knowledge are interpreted as evidence of alien interventions. This approach ignores the principle of Occam's razor, according to which among different possible explanations, the one requiring the fewest assumptions is preferable. From a genetic perspective, theories of alien intervention find no support in modern scientific research. Non-coding DNA, far from being an inexplicable mystery, is gradually revealing its functions through rigorous studies. The peculiarities of human evolution, including brain development, can be explained through known evolutionary mechanisms, such as natural selection, genetic drift, and adaptation to specific ecological niches. As highlighted in Boschetto's document, The Theories of Ancient Astronauts, the hypothesis that humans were created by technologically advanced extraterrestrial intelligences remains in the field of speculation, without concrete scientific evidence to support it. The scientific community considers these theories as pseudo-scientific, as they do not follow the scientific method and do not provide verifiable evidence to support their claims. This does not mean that research on the possible existence of extraterrestrial life is not scientifically valid. On the contrary, fields like astrobiology and SETI, Search for Extraterrestrial Intelligence, research use rigorous scientific methods to explore the possibility of life beyond Earth. However, these disciplines are based on empirical evidence and testable hypotheses, clearly distinguishing themselves from unverifiable speculations about ancient astronauts. Theories about ancient genetic engineering by extraterrestrials thus represent more a cultural phenomenon than a real scientific hypothesis. Their charm lies in the ability to offer simple answers to complex questions about our origins and our uniqueness as a species. However, the true story of human evolution, written in our DNA and in fossil remains, is probably more complex, but no less fascinating. In the debate on the origins of non-coding DNA and possible extraterrestrial influences on the human genome, It is fundamental to distinguish between scientific approach, theories at the boundary of science, and pseudoscience. This distinction is not always clear-cut, and there are often gray areas where speculative hypotheses may eventually evolve into scientific theories or be definitively refuted. The scientific approach to non-coding DNA and the origins of life is based on some fundamental principles: the formulation of testable hypotheses, the systematic collection of empirical data, peer review, and the willingness to revise theories in light of new evidence. Science proceeds by successive approximations, gradually building an increasingly accurate picture of reality. In the case of non-coding DNA, scientific research has progressively abandoned the idea that it is junk DNA devoid of function to recognize its crucial role in gene regulation, chromatin structure, and numerous other cellular processes. This paradigm shift did not occur through unverifiable speculations, but thanks to rigorous studies that provided concrete evidence of the functions of these genomic regions. Similarly, the theory of panspermia, while considered at the boundary of mainstream science, is based on some empirical observations. The resistance of certain microorganisms to space conditions, the presence of complex organic molecules in meteorites, the theoretical possibility of material transfer between celestial bodies. These observations do not definitively prove the theory, but make it a scientifically discussable hypothesis, albeit still controversial. The pseudoscientific or speculative approach, represented by theories about ancient astronauts and alien genetic engineering, is characterized instead by unfalsifiable claims, selection of data that support a preconceived theory, publication mainly in channels not subject to peer review, and resistance to modifying theories in the face of contrary evidence. As highlighted in the CICAP article, supporters of these theories tend to interpret archaeological artifacts without considering their cultural context, to select only the examples that seem to support their view, and to use the argument from ignorance as proof of their claims. This methodological approach is fundamentally different from the scientific one, which requires rigor, transparency, and falsifiability. However, it would be simplistic to completely dismiss the value of speculation in scientific progress. The history of science is rich with examples where ideas initially considered heretical or fanciful later proved correct. The theory of continental drift, proposed by Alfred Wegener in 1912, was initially ridiculed by the scientific community, only to be accepted decades later with the advent of plate tectonics theory. The crucial difference lies in the fact that these ideas, to be eventually accepted as scientific, must evolve from pure speculations to testable hypotheses, supported by empirical evidence. Speculation can serve as a starting point for the formulation of new hypotheses, but it cannot replace the rigorous process of empirical verification that characterizes the scientific method. In the specific case of non-coding DNA and its possible origins, there are some points of contact between scientific hypotheses and speculative ones. Both approaches recognize that there are still many aspects of the human genome to understand, and that the question of the origin of life on Earth is far from resolved. Moreover, modern science considers the existence of life elsewhere in the universe plausible, albeit with approaches very different from those of ancient astronaut theorists. The fundamental divergences concern methodology, the principle of parsimony, and the progressivity of theories. The scientific approach requires verifiable evidence and falsifiability, prefers explanations that do not require the introduction of unnecessary entities or processes, Occam's razor principle, and is characterized by the ability to evolve and refine with the accumulation of new evidence. A concrete example of this methodological difference concerns the interpretation of non-coding DNA. While science has gradually discovered specific functions for many of these genomic regions through rigorous studies, supporters of alien intervention have simply postulated that these sequences might contain codes inserted by extraterrestrial civilizations without providing verifiable evidence to support this claim. In conclusion, in evaluating the different theories on the origins of non-coding DNA and possible extraterrestrial influence on the human genome, it is fundamental to distinguish between scientific hypotheses based on empirical evidence and rigorous methodologies, and unverifiable speculations. This distinction does not necessarily imply that alternative theories are devoid of cultural or intellectual value, but recognizes that their epistemological status is fundamentally different from that of scientific theories. At the end of this journey through the frontiers of genetics, evolutionary biology, and astrobiology, we find ourselves facing a complex landscape where consolidated scientific knowledge intertwines with still open questions and speculations of various nature. Non-coding DNA, once dismissed as evolutionary junk, has revealed itself to be a rich and complex territory, whose functions we continue to discover and understand. Modern science has made giant strides in understanding the human genome. We know that non-coding DNA plays crucial roles in regulating gene expression, chromatin structure, cellular stress response, and numerous other biological processes. Non-coding RNAs transcribed from these genomic regions constitute a vast and diverse class of regulatory molecules, whose importance has been recognized only in recent decades. However, it would be presumptuous to claim that we have solved all the mysteries of our genome. Vast regions of non-coding DNA remain enigmatic, and their function, if it exists, is yet to be discovered. The evolution of these sequences, their conservation in different species, and their contribution to the complexity of organisms are questions that continue to stimulate scientific research. The theory of panspermia, while not solving the problem of the primordial origin of life, offers interesting insights into the possibility that the building blocks of life may be spread throughout the universe. The evidence of the presence of complex organic molecules in meteorites and comets, and the resistance of some microorganisms to space conditions, suggests that Earth might not be an isolated biological island, but part of a cosmic network of organic material exchange. Theories about alien genetic interventions, while fascinating from a narrative perspective, lack solid scientific and methodological foundations. They represent more a cultural phenomenon than a real scientific hypothesis, reflecting our ancestral desire to understand our origins and our place in the universe. As Carl Sagan observed, extraordinary claims require extraordinary evidence, and so far such evidence has not been provided. This does not mean that the question of extraterrestrial life is scientifically invalid. On the contrary, the search for life beyond Earth is a rapidly expanding scientific field, which uses rigorous methods to explore the possibility that life may exist or have existed on other celestial bodies, such as Mars, Europa, or Enceladus. The crucial difference lies in the methodological approach. Science proceeds through testable and verifiable hypotheses, not through unfalsifiable speculations. The enduring fascination with alien origins of humanity probably reflects a deeper truth. We are indeed children of the stars, in the sense that the elements that make up our body, carbon, nitrogen, oxygen, and all the others, were forged in stellar furnaces and dispersed throughout the universe through supernova explosions. As astrophysicist Neil deGrasse Tyson poetically observed, we are all connected, to each other biologically, to the Earth chemically, to the rest of the universe atomically. The future prospects of genetic and astrobiological research are exciting. New DNA sequencing and analysis technologies are revealing increasingly fine details of the structure and function of the genome. Space missions like the Perseverance rover on Mars or future missions to Europa and Enceladus will search for traces of past or present life. The James Webb Space Telescope and its successors will scrutinize the atmosphere of exoplanets in search of biomarkers. In this context of rapid scientific progress, it is important to maintain a balance between scientific rigor and openness to the unknown. Science proceeds through methodical doubt and empirical verification, but also through the ability to imagine possibilities not yet explored. As physicist Richard Feynman wrote, "The pleasure of finding things out, the joy of discovery, is certainly the greatest that the human mind can experience." In conclusion, the mystery of non-human DNA in humans, or rather of the vast non-coding regions of our genome, remains a fascinating field of inquiry in which science continues to make surprising discoveries. The origins of life on Earth and the possibility of life elsewhere in the universe are questions that continue to stimulate both scientific research and human imagination. In this dialogue between knowledge and mystery, between what we know and what we can only imagine, perhaps lies the true value of the search for our origins.